What determines hair color, or skin color, or fur color in animals? Obviously, it has to be something genetic, right? Genes code for proteins, and proteins are the molecular machines within cells that are responsible for almost everything that cells do, from fighting disease, to turning the food that we eat into energy, to producing the pigment that we find in hair or in skin. As you may suspect, there isn't one single gene that's responsible for hair color. In fact, there are more than a dozen different genes that have been identified so far that play some role in the processes that ultimately produce the pigment that you find in hair. Now, this is actually pretty common. Most traits in biological organisms are controlled by a whole suite of genes. In humans, think about height. There's no tall gene or short gene. Think about intelligence, or voice pitch, or body shape, or bone density. Any number of traits that humans have are controlled by 2, 3, 4, up to several dozen different genes. In this video, we're going to examine the genetics of mouse fur color in the mouse Peromyscus polionotus. This is one of those traits that's controlled by a whole host of genes. It's actually a pretty complex picture. But we're going to focus on the nucleotide sequence of one specific gene that controls somewhere in the neighborhood of 10% to maybe 40% of the variation that you see in mouse fur color. This gene is called MC1R, or the melanocortin-1 receptor gene. We'll explore how one tiny mutation can have pretty interesting consequences when it comes to mouse fur color. The melanocortin-1 receptor gene is on chromosome 16 in mammals. The RNA coding portion of the gene is 951 nucleotides long, and it codes for a string of 317 amino acids. A protein, of course, is merely a chain of amino acids. So those 317 amino acids, those make up the MC1R protein. Now, how do we know that this gene has anything to do with mouse fur color? Well, there was a team of researchers from Harvard, led by Dr. Hopi Hoekstra, and they decided to tackle this very question. Peromyscus polionotus is interesting in that there are several different populations that inhabit different beaches all around the coast of Florida. Because of human development over the past couple hundred years, these populations have been isolated from each other for quite some time. One thing that tends to differ from one population to the next is the color of the fur on the mice. So, Dr. Hoekstra and her lab took genetic samples from individuals in each population and sequenced the MC1R gene. They could then explore whether or not the sequence of nucleotides in the MC1R gene differed between mice in light versus dark populations. What they found was actually quite interesting. The nucleotide sequence in the MC1R gene for mice with dark fur versus mice with light fur was identical, except for the one nucleotide that was located at position 199 of the 951 nucleotide sequence. For mice with dark fur, that nucleotide was usually cytosine. For mice with light fur, that nucleotide was usually thymine. Okay, so what difference would that make? Just a single nucleotide out of 951. Well, if that nucleotide was cytosine, then the corresponding codon would code for the amino acid arginine. But if that nucleotide was thymine, then the corresponding codon would code for the amino acid cysteine. All right, what difference would that make? We changed just one amino acid out of 317 amino acids that make up the MC1R protein. Well, let's take a look at these amino acids. On one hand, we have arginine. On the other hand, we have cysteine. Arginine is a significantly larger molecule than cysteine. It's about 50% larger by molecular weight. Not only that, it's positively charged. This would cause it to repel or push away from other positively charged amino acids within the protein. It would also be drawn closer to or attracted to negatively charged amino acids. The positive charge also makes it super hydrophilic or attracted to water. Cysteine, on the other hand, is smaller and it's also non-polar, meaning that it wouldn't be particularly attracted to or repelled with respect to other charged amino acids. It's also not attracted to water. You'll note that it contains the element sulfur. Sometimes sulfur in one amino acid can react with the sulfur in another amino acid within the protein and form what's called a disulfide bond, which helps to give the protein its unique shape. Now you can imagine if there was a mutation that resulted in a cysteine being built into the MC1R protein instead of arginine at one particular location, that could have some pretty significant consequences for the shape and the function of the protein. Hoekstra and her colleagues were actually able to quantify, to some extent, how much of an impact this tiny mutation had on mouse fur color. They did a genetic analysis of more than 450 mice. For each mouse, they determined whether it was homozygous for the allele that produced the arginine version of the protein, homozygous for the allele that produced the cysteine version of the protein, or if the given mouse was heterozygous, meaning that it would have one copy of each of the two different alleles. For mice that were homozygous for the allele that produced the arginine version of the protein, all 126 of them had a dark brown fur color. For the 215 mice that were heterozygous for the MC1R gene, that means they had one of each type of allele, 81% of them had dark brown fur, 19% of them had medium brown fur. Finally, for the 118 mice that were homozygous for the allele that produced the cysteine version of the protein, 41% of them had light brown fur, 36% of them had medium brown fur, and only 23% of them had dark brown fur. Now remember, most traits are controlled by more than one gene. Fur color is no exception, as I mentioned at the beginning of this video. MC1R is only one of a few dozen different genes that contribute to the production of hair pigment. But what seems to be clear from Dr. Hoekstra's work is that this simple single nucleotide mutation in the MC1R gene has a significant and measurable impact on the fur color in the Peromyscus polionotus mouse. To complete the picture of why and how the MC1R gene is associated with mouse fur color, be sure to check out the mouse fur color genetics content on our EvoEd website. To explore a different aspect of this case, you can follow the links on the website or down below in the description. Thanks for tuning in. I'll see you next time.